Hello, everyone. Welcome to the CARC data facing track call for July. My name is Deb McCaffrey. And just for some of the old hats here, we're trying to make our calls more accessible. So first thing to that regard, so I'd like to describe myself and my surroundings a bit for visually impaired folks. So I'm Deb McCaffrey. My pronouns are she, her. I am a blonde-haired white woman wearing a green shirt, and I have a virtual background on of a scene from Doctor Who. And before we get started, on your screen right now is just a slide about the entirety of CARC. It is a professional organization dedicated to what we call research, computing, and data. On the screen, we have a graphic showing all of our different activities and kind of when they started, if any of them ended. Then on the right side, we describe what our people network is. The data facing track is a part of that. This is kind of our year-round remote conference. We have a data facing track, emerging centers track, researcher facing track, and systems facing track. And then the other half of CARC is working in interest groups that are activities around deliverables. We have a professionalization and career arcs, capabilities model, engagement, the RCD Nexus, EPSCoR, CI workshop, and the decadal survey. So that's just a little bit about everything that's going on. If you would like to know more about anything, feel free to contact us after the talk. And with that, I will introduce our speaker. This is Marisa Conti. She's the Associate Director of Research and Informatics at the University of Michigan's Taubman Health Sciences Library. In this role, she facilitates library collaboration in research and informatics initiatives. Marisa is also a PhD student in health infrastructures and learning systems in the University of Michigan Med School's Department of Learning Health Sciences. Her research interests include infrastructure for the management and dissemination of computable biomedical knowledge. So off to Marisa.
Great, thank you very much. As I get my slides set up here, I will describe myself very quickly. I am a white woman, probably very red-faced at the moment because it's about 900 degrees in the office where I am currently working. My background is a blurred out office. I have a window that faces onto a central corridor. So in the interest of protecting the privacy of my colleagues who may not expect to be in a recorded meeting, I do have that blurred out. I am going to be sharing a screen, and hopefully at this point that is visible. Deb or Amy, yours are the only names I can see in the sidebar at the moment. Would you be willing to give me a thumbs up?
Yeah.
Great. And I also will not be able to see the chat really readily. So if either of you could please feel free to interrupt me if there's questions coming in as I am presenting.
Will do.
Thank you so much. And thank you all for having me here today. I was asked to talk specifically about common data elements. And what I would like to do is really weave that into a broader talk about standardizing data specifically for use in clinical research. And I know as folks were signing on, there's a lot of awareness of what's coming in terms of policy with the new NIH data sharing and management policy that will take effect in January of 2023. So while that is not the specific focus of the talk, as I talk about common data elements, one thing that I hope to do is give you some resources and build awareness about where you can find and access elements and help your researchers to use these in their work. So this will be a little bit of repetition, but I'm going to go ahead and introduce myself. Again, my name is Marisa Conti. I've got some contact information on the slide here. Most importantly, my email, which is the easiest way to reach me, is meese, M-E-E-S-E, @umich.edu. And I'm here with kind of two hats on. So on the one hand, I am the Associate Director for Research and Informatics at the Taubman Health Sciences Library. I lead a really engaged team of 7 individuals who integrate into the biomedical research enterprise at the University of Michigan. And obviously, data is something that we're spending a fair bit of time on these days. And then as Deb mentioned, I'm also a PhD student. If I were doing this talk 2 months from now, I would hopefully be able to say PhD candidate, but we're not there yet. At the University of Michigan, the medical school has a Department of Learning Health Sciences. And I've put one of the elements of our program is this aspect of a learning cycle, which really looks at how knowledge and data circulate through health systems, whether that's a clinical setting or whether that's a research setting, beginning with the formation of a learning community that identifies a problem, looking at performance, translating performance to data, data to knowledge, and then knowledge to performance again in this virtuous cycle that is supposed to help identify, fix, and then continue to learn from the operations of the health system or the research context that they're working in. And my interest is really how do we look at something like computable biomedical knowledge that's generated from these large amounts of data that we have either from research settings or from health contexts, and be able to manage and disseminate that more effectively than we're doing right now. So what kind of infrastructure is needed for that, and also what sorts of social infrastructures are needed to make that happen. So with that in mind, standards are a large part of what I'm interested in and the things that I work with. And we'll talk a lot about standards through this presentation, but I wanted to start off just by setting the stage. I understand that not everyone here works in health sciences, that we have people that are in libraries where you're handling lots of different data from lots of different disciplines. So I wanted to start off just by setting the stage for what I would be talking about today. And I think one of the things that's really important to do in health research is to distinguish between research data and then clinical data on which research is done. So I'm distinguishing here between research data and then the secondary use of clinical data for research purposes. So on the slide here, there's a little graphic of an individual. There's pills in the lower right-hand corner, which are meant to represent some kind of experimental intervention. And then up over the person's shoulder, you see a bar graph, which is meant to represent the data that's coming from this study. So when we talk about research data in this very general sense, we're talking about data that's collected for a specific experimental purpose. So that could be a clinical trial, it could be a longitudinal study. And often with these sorts of studies, you're using things like standardized and validated instruments. You're using case report forms to collect the data from the individuals that have agreed to participate in your study. And it's a pretty standardized environment compared to a clinical environment. So what you see here is a patient receiving care, laying in a bed. There's a care provider standing over the bed and a little heart that signifies that care is being provided. There is a fair bit of research that's done using clinical data, and we call that secondary use of clinical data. This can be also known as real-world data. And this is where data is derived from an electronic health record or from some of the other administrative sources or data warehouses that are linked into an electronic health record. This can also be used in research. So you can see this used in retrospective studies or observational studies. And it can also facilitate prospective research. So EHR-derived phenotypes can be used for trial cohort recruitment. And what that means is somebody can write an algorithm that will operate over an electronic health record to select all of the patients that have a particular phenotype, which is basically just the characteristics of interest for a particular study, and then be able to identify a cohort much more effectively than, in some cases, much more effectively than using traditional recruitment methods. The other thing that I want to distinguish between is data that are human readable versus data that are machine readable. So we don't want humans to come completely out of most processes to include research processes, but there are some benefits to having data that can be machine readable in terms of being able to collect data, to be able to analyze data, and to be able to disseminate data. And that's another value to having standards in place, is that they can actually be machine readable and can help to do things at a much larger scale than humans can do alone. So when we talk about common data elements being used in research, we're talking about them for a couple of different reasons. This is an audience that I expect I do not have to explain what FAIR is. That stands for findable, accessible, interoperable, and reusable. And standards such as common data elements are one way to help make data more fair so that it can be used by more people for more things across a very broad spectrum. It can also make the data more reusable and increase the potential uses of this data. So the text on this slide comes from the HEAL initiative, which we'll talk about in a few more slides. It's an NIH program that addresses addiction. And they have a common data element that is used across all of the sites for this particular study. And they have a very nice website that talks about the benefits of using a common data element. which include, for this study, the ability to compare interventions. So because different sites are going to be collecting different information on larger or smaller populations, it gives a nice basis from which these interventions can be compared across multiple sites. They cite as a benefit an access to a source of preclinical data. So again, because the data are being collected in a standardized manner, instead of having to refer back to animal models, you can actually use the human participants who have agreed to participate in the study as a source of the preclinical data. It gives a better understanding, a deeper and richer understanding, because again, you're able to aggregate data from so many different contexts in the same way, and then ultimately leads to a larger evidence base, which can hopefully facilitate discovery more efficiently. Using common data elements and standards can also help to affect research at scale. So this is actually an example of a common data model, which is different to a common data element. But I just wanted to, I like this slide because I think it really does tell the scale story. So this is within a clinical data research network and shows that they started with about 650,000 participants. And through the process of being able to identify, approach, attract patients to a portal, have them complete a set of questionnaires on the portal, and then being able to actually identify eligible patients, they were able to randomize about 15,000 out of this very large population. So if you think about clinical research and what it would take to actually be able to go out and screen 650,000 people to identify a cohort of 15,000, being able to do some of these in a little bit more of an automated process really makes sense, and you can see why standards would help with that. And without standards, really, we have a couple of different problems. So we have the mystery box problem. As you can see here, you've got a box. It probably has some cool stuff in it, but without a way to describe what's there, we don't really know what's in the box. Standards can help with that. Standards can also help when people have identified the same thing, but are talking about it in two different ways. If we can actually have people speaking the same language when it comes to some of the things that are being collected or measured, it can help to facilitate research. And without standards, it is possible to map between different things that have been collected, but there's a couple of risks here. You have a risk of definitely losing information as things are mapped from one setting to another. And another problem with this is that it's time and resource intensive, and usually that time and those resources can be better spent doing other things. So with that basic context, the difference between data collected for the purposes of research and clinical data that's used in secondary purposes for research, what I will be talking about today are common data elements. So that's what I was asked to speak about. So that'll be the bulk of the talk. I will talk a little bit about terminologies and standards because the common data elements often refer back to terminologies and standards to be able to handle things like drug names or lab tests or vital signs reporting in a consistent way. And then I will briefly mention common data models. And I think the simplest way to think about this is that a common data element is something that's used at a researcher level. A common data model would be something that would be potentially be used at a study level or at an institution level. And there are different ways of handling data. What I won't be spending a lot of time talking about is the common data models. So that would be another talk in and of itself, and I won't be going into a lot of detail on those. I won't be talking about reporting standards. So there are a lot of standards out there for adverse event reporting, or the FDA has specific requirements and standards for how data are reported. I will not be talking about the Clinical Data Interchange Standards Consortium, CDISC, which hosts and produces C-, which is a standard for clinical data acquisition and harmonization. I will not be talking about the FHIR resources, the Fast Healthcare Interoperability resources. And I just wanted to put all of that out there because if there are people that were hoping to be able to hear about some of those things, just so you know that that's not going to be covered in this talk. And also, unfortunately, we're not going to have time to go into a lot of detail about any specific things. I will do my best after the talk to answer questions with the caveat right now that I am not a clinical researcher nor a clinical coordinator. So some of the nitty-gritty questions might be a little bit outside of my area of expertise. So starting off with a couple of definitions. What is a common data element? And the first definition is coming from the NIH Common Data Elements, or CDE repository. And this site defines a common data element as a standardized, precisely defined question that is paired with a set of specific allowable responses that is then used systematically across different sites, studies, or clinical trials to ensure data collection. And I've bolded out the things here that I think are really the salient points of this definition. The fact that it is standardized and very precisely defined as a question. And then also the fact that it's actually paired with the responses that are allowed. So in a real simple way, you can think of that as this is the difference between maybe a validated instrument and a common data element, is that the common data element is actually going to come with the answers bundled in, which makes it easier for coding, for scoring, for analyzing, things like that. And then the fact that this is common data elements are intended to be used systematically across different sites, studies, or clinical trials. So they aren't intended to be one-offs. They really are intended to be something they're going to have community use to be able to deliver benefit back to the community. And the second definition also comes from NIH and again from the HEAL initiative, which we'll talk about in a few moments. And this study defines CDEs as defined fields describing the data to be collected, for example, identifying specific variables, along with how to gather the data and how the response is represented in a data set, and specifies that CDEs are structured as indivisible units of data. And the thing that I like about this definition is it kind of gives you the who, what, where, when, why, how, in that you're describing what you're collecting, which is the data, how to gather the data, and then how the response is actually going to be associated with the data for a particular question. So just as a quick example, these are 3 different ways of measuring pain. And just in looking at this, you can see where if you were trying to aggregate data from studies that had used each of these scales independently, you could see where it may be very difficult to come up with what was going to be your unit of analysis. So you have in the top corner, you have a 0 to 10 numeric pain rating scale, with 0 being no pain and then 10 being the most severe pain. Below that, you have a color-coded set of faces. Some of the faces are very happy and some of the faces are very, very unhappy. And again, you're starting with no pain on one side and running up a scale to hurts the worst. And then the most complicated example shows the image of a human front and back, which is where someone could indicate where their source of pain was. It has in 2 columns a number of different words that would be used to describe the pain. Some of these are how you would actually describe the physical sensation, and some of these are how you might emotionally or mentally process the sensation as you're experiencing it. There is an indication for internal or external in terms of the source of the pain, and then in terms of the duration, there's little boxes to check that run a scale from brief to constant. So again, these examples are here just as a very common thing that might be collected in the course of a clinical study to show that there are many different ways to do it. You could make an argument for why any of these would be the most valid for a particular study, but understanding that without using some common data element, it's going to be very difficult to use those in any kind of comparison or group them with another study in any way. Before I move on from this slide, I'll just point out at the very bottom, the NIH-CDE site does have a nice little tutorial. That's actually what most of these images have been taken from. So if you're interested in learning more about common data elements and have the time to work through a quick tutorial, I would refer you to that. So on to some of the sites for common data elements. We are actually going to be starting at the common data element homepage. So the URL is at the top of the page, cde.nlm.nih.gov. So like many of the resources that we'll be talking about today, this is produced and maintained by the National Library of Medicine, which is one of the National Institutes of Health. And what the CDE repository does is it aggregates common data elements from different NIH ICs, institutes and centers, as well as other organizations that have contributed. Just really quickly across the top, you have places that you can click for CDEs, which is where you can search or browse the common data elements. Forms, which are pre-aggregated CDEs that address a specific topic. So I'll have an example of that in a moment. My boards, which we'll talk about, which is where an investigator could actually go through and pick out common data elements that they would use to describe or to integrate into their instrument, and they could assemble a form basically from this repository. And then they've got some help features. There's a little yellow ribbon on NIH-endorsed CDEs, so I wanted to talk a quick moment about this endorsement process. To the best of my understanding, this actually only applies right now to some CDEs around COVID. So the endorsement is actually a consensus process, and there's a governance committee that looks at CDEs and holds them up to criteria that measure the definition, documented evidence of reliability, whether it's present in both human and machine readable format, whether there's licensing information available, and whether it's been recommended or designated by an NIH body, like an institute or center, a trans NIH committee or something like that. So as you're going through, as time goes by, maybe we'll see more of those that have the little gold ribbon on it, but that's just another indication of the efforts that NIH is making to really endorse the FAIR principles as much as they can. So back on the, so what I've done is from here, if I scroll down on this page, you get to a part of the website that has three little boxes. One says explore, one says learn, and one says submit. And what I've done here is click on the explore so that you can see the drop down. And this is how you actually get in and start looking at what is in the Common Data Elements repository. There are search features where you can put in keywords. There's a browse feature, which I'll show in a moment, where you can browse by institute and center to see what's there. The next box has a learn, and this talks about what common data elements are and how they can be used to support data sharing. There is a tutorial. So I mentioned earlier that I had taken some images directly from the tutorial. That's here. And then again, just a great resource if you want to learn more about it. I didn't have anything in this presentation for submit, just because I think it's a little bit outside of our scope. The last thing that I need to mention before we go any further, on all of the preceding slides, you've seen a little sign-in button up in the corner. And this site is managed by the same authentication process as another NLM hosted product, which is called the Unified Medical Language Service, system. And you do have to sign up for an account to be able to actually see the different elements within a common data element. So you can only get so far with the search, and you'll get to a point where it'll show you in a table view what common data elements are there. But if you want any additional information about most of these common data elements, you're going to have to log in. And the login process, the simplest thing that I found is to go in through your institutional credentials. I set that up once, and it actually is much easier than trying to remember a separate NIH login or something like that. This license is actually for the UMLS. So you will be setting up an account with the UMLS. So there'll be a lot of language that you'll have to read through that talks about what you can and can't do with the source vocabularies within this UMLS system. But again, if you want to see specific information about the common data elements, it's really the only way to do it. So this will be a required step of the process. So once you've logged yourself in so that you'll be able to see everything, the first way that you can look for terms or common data elements is to browse by the sponsor. So we see the Project 5 COVID-19 has the little yellow ribbon on it. That's the endorsement process that I mentioned earlier. But if I just read along this row in the table, I can see that the Agency for Healthcare Research and Quality has some common data elements here. The National Cancer Institute has some common data elements here, as well as some of the other NIH institutes and centers, like the National Heart, Lung, and Blood Institute, Institute of Nursing Research, and so on. So if you're going to be working with researchers that are starting to think about a data sharing or a data management plan to meet the policy, perhaps one thing that you could do would be to go into the Common Data Elements repository, browse by sponsor, and see if you can find any elements that might be relevant to that particular study. The researcher could then write into their data sharing plan that they will be using common data elements and talk specifically about what the source of those are and what they would hope to accomplish by doing that. The other thing that you can do is a search. And what I've done here is I've just done a very simple keyword search for the word pain, looking perhaps for pain scales or things like that. And then I've copied and pasted some different aspects of a big page of results that you get. So the first thing is down the margin of the page, what you're going to see is an opportunity to filter down a little bit. So if I just do a general keyword search for pain, and what I really want is maybe I want pain from National Cancer Institute, I can see right here that there's going to be 20 that are going to be relevant. I can click there and then be able to specify further. It just helps me to filter down. What you see in the center panel, because I've signed in, you actually see a name of a common data element. You see the values that could be used in answering that, the code name that actually describes the values for the humans that are going to be reading this, and then the specific codes. And because a lot of these codes come from proprietary sources that are licensed through the UMLS, that's the reason that you can't actually see these until you're logged in. And what we can see here, this is just a quick screenshot that I grabbed. One is just a general chest pain, one has pain type, and then one is a pain behavior when went scale, which is part of a really detailed patient-reported outcomes measure series. This is a view from inside of a common data element record. So what I clicked on was a common data element. And again, these are very specific. So this is a record element for in the past 7 days, I limped because of pain. And they've got a scale. There's a table that has a value from 1 to 6, with 1 being had no pain, 6 being always had pain. And then the more information that you want, you can see what the source code system is, which in this case is LOINC, which we'll talk about a bit later in the presentation. Along the left-hand side, you can see all of the aspects that are going to be covered by a pain record or by a CDE record. So you can see everything from the general details down to classification. It'll actually link out to reference documents where those are available, talk about history and things like that. All right, so we are now moving on to forms. And I had mentioned at the very top of the home screen that you could click on CDEs and you could click on forms. The difference between a CDE and a form is that a CDE is just going to be one element. So it's just going to be one question with a set of allowable answers for that question, whereas a form is going to bring together a lot of different CDEs related to a specific topic. So what I did here was, again, I just did a search for pain in the forms, and there's 2 different views, and I wanted to show both just so you can see the different levels. So the summary view is kind of like what we looked at when we were just looking at the common data elements. So it gives you all of the different common data elements that are available. And then along the left-hand frame of the page, you'll have some options to be able to refine down by specific facets. In the table view, if you click into the table view, at first all that shows up is the name and it doesn't look particularly helpful. But then when you open the table view fields, you can see that you can actually pick out quite a few different elements to be able to filter by. So you can actually look at the number of questions in the table view. And if you know specifically or more specifically what you're looking for, perhaps the table view is a little bit quicker way to get you there. And this is an example of what a form might look like. So this is from a research group called Phoenix. They're looking at pain caused by toothache. And you can see that they've got one question about describing the pain, one question asking about whether this happened more than once, one question that asks about rating the pain on a pain scale. So that's just an example, again, of how a form is going to bring together more CDEs related to one question. And sometimes if you can find a form, maybe that would be an easier way to go than picking out individual elements. But on the off chance that you feel like picking out individual elements, there is a way to do that too. And if you remember back to when we looked at the home screen, there was a link for My Boards. And that's where you can actually, once you're signed in, identify individual elements or individual forms and be able to put things together for yourself. So within an element view or a form view, you'll see a little pin, a little push pin icon that says add to board. And that's how you would start to create a board for yourself. If you're within a form and you only want certain elements out of that form, if you click the pin CDEs, it actually extracts the individual elements from the form and adds those to your board. I wish I trusted my internet connection enough to do a live demo here, but I didn't. So that's why we're working with screenshots. Before I move on to some of the other sources for common data elements, because the NIH-CDE is great, but it's not the only thing that's out there, I did want to point out one potential weakness to the NIH-CDEs at the moment, which is that it's not used by all of the institutes and centers. So there's about 30 institutes and centers at the National Institutes of Health. And if we look here, there are not 30 different participants here, and some of them aren't even NIH in particular. So what this means, if we're going to be working with researchers to look for sources for common data elements, is I think it's really reasonable to make the NIH CDE repository your first stop to see what's available, but I would not stop there. And we'll talk now about some other resources that are available. One of the resources is going directly to the institute or center that is sponsoring the research that the investigator you're working with is writing a grant for. So the National Cancer Institute has its own CDE browser. I've tried this in a couple of different browsers, and Firefox is the browser in which it has by far worked the best for me. So if you're going to be using it, if you log in and the tabs are all stacked on top of each other, it's not necessarily a problem with the website. I would maybe just switch your internet browser and try again. But this is just an example of how a pain inventory will appear in the NCI CDE browser, which does have, I don't think that there's a one-to-one overlap between the content that's in the NIH CDE browser and what's available in the NCI. So one thing that you might want to do again is go to the institute and/or center that is sponsoring the research and take a look there. Another thing that you might want to do is just take a look at the specific project. So if the project has its own website, there might be some information there about what common data elements are required and how they're required to be used. So we had talked earlier about the HEAL initiative, which stands for Helping to End Addiction Long Term. This is a big NIH initiative, and they're actually fairly prescriptive about how common data elements have to be used. So they specify a set that have to be used. This is just a subsection that I could grab on a screenshot of some of the elements, CDEs that are required for use. And then they've got an entire hierarchy of how you could actually, if you want to use an instrument that isn't part of what's being used already, how that has to be vetted, just to make sure that the data that are being collected are going to be as consistent as possible across this gigantic multi-site study so that they can be as reusable as possible. One thing that I like about this is they list out not only the dimension that they're measuring with the common data element, but they describe it and then they actually link out to the source documentation. So I think that for investigators who wanted to really understand specific elements and where it would be helpful to use these, I think that this is a great resource. This is another example, and this is a collaboration actually between NINDS, which is the National Institute of Neurological Disorders and Stroke, and then a professional society, the American Academy for Cerebral Palsy and Developmental Medicine. And what they've done here is put together a set of common data elements for children who were not represented in the original common data element set. What I liked about this, and the reason I included this, is that it actually walks through the process. So at the top level, there's a bar that shows the NINDS CDEs, the actual domains that are used for cerebral palsy. They break out into smaller boxes, including participant characteristics, health growth, genetics, comorbidities and labs, neuroimaging, all the way to engagement and quality of life assessments. And then it shows how they've actually narrowed down to create these, the CDEs that are used for children. They have working groups. The working groups then fed down to an internal review group that sent things out for public feedback, that got additional input from stakeholders that then led to final revisions. So I think the transparency and the number of people that were involved in setting up this element set is really interesting. And I've linked to the article at the bottom. I'm presuming these slides will be available to you at some point. This is an open access article. So if you wanted to kind of go behind the curtain and see how this was created and the reasoning for putting one of these together, this is a good resource to use. And then professional societies will sometimes have their own common data elements library. So this is ISTH, which I have completely forgotten what that stands for. I think it's thrombosis. But they have a common data elements library. So again, if you've checked the supporting NIH-IC and they don't seem to have common data elements, if you've looked, if your investigator is writing into a larger study and they don't seem to have common data elements, then perhaps looking at some of the professional societies or organizations that are going to be aligned with the type of research could be a way to find some common data elements that could be used. And this goes beyond health data. So what I want to talk about next goes, this might be a little more of an overlap between some of the data gathered for research purposes and the clinical data used for secondary research purposes. But there's a group called the Gravity Project that is creating common data elements for social determinants of health. And this is going to obviously impact a lot of research studies. So if you have researchers that are going to be collecting not only demographic information, but maybe they want more information about social determinants of health that could be impacting the volunteers or participants in their research, this project is definitely worth keeping an eye on. So it's a big collaborative organization. They've got stakeholders from academia, from industry, from patient advocacy groups, and they divide their work into 3 different work streams. So the one that's been represented here is the terminology work stream. And in the terminology work stream, sorry about that, in the terminology work stream, you can see the little colored dots on this slide, and these all represent the different social determinants of health for which this group is developing common data elements, and they're developing them across 4 different fields. And on this slide, you can see one that's actually been completed. So the 4 fields that they're developing these SDOH, social determinants of health, common data elements across are screening, diagnoses, goals, and interventions. The only one that they finished completely at this point is food insecurity. And you can see that because all of the boxes are checked in the comprehensive list of codes present. You can click on this link and you can actually get out to the spreadsheet that shows you what is going on within these common data elements. So this one here just shows an example of so hunger vital signs, different places that have contributed content to this common data element. This is a website that you don't need to log in for. So their website is a regular website, and then they have a wiki through HL7 that's hosted on a Confluence site, and you can see all of their materials at any time on that site. Before I head into terminologies, actually, I'm going to show you where they matter, and I'll just use this slide because it happens to be the one that's closest. So again, we're back at the table out of the Gravity Project, and the table has a number of columns. The first one is a screening tool. Then they've got a panel name, question concept, answer concept. And through all of this, you're seeing LOINC, 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 LOINC. So LOINC is the terminology that's actually being used here. And in some places, what you're getting is the actual text that's going to be coming out of LOINC, and in some places you're getting what an answer would code to. So if we look at those last 2 columns, you have LOINC answers up in the first row of often true, sometimes true, never true, don't know, or refused, related to a question about whether the person has worried in the last 12 months about food running out before they had money to buy more. And then the column next to that is the LOINC code that actually pairs with that textual answer, and that would be the machine readable part of it. So LOINC is a terminology, and the terminology itself is used as part of the data element to be a standard use of a specific concept. And in terms of terminologies, I've just put probably the ones that you're going to see most frequently in common data elements on this slide. So starting at the top, RX norm is a vocabulary or a standard that's produced by the National Library of Medicine, and it's used to normalize drug names. So what that means is it's going to bundle together generic names as well as trade names, and then have a lot of information, all of which can be encoded for use by computable systems about dosage and different things like that. SNOMED CT, it stands for Systematized Nomenclature of Medicine, and this is a pretty comprehensive health terminology. It's actually used internationally. LOINC is the one that we were just talking about on the previous slide. So that's our logical observation identifiers, names, and codes. So this is where we get a lot of our standardized values for lab tests, the values, vital signs, clinical operations, observations, and things like that. And that's produced by the Regenstrief Institute in Indiana. ICD-10 you might see, so that's a coding system, International Classification of Diseases. So we're up to ICD-10 finally in the US. This is maintained by the World Health Organization, and within the United States, use is overseen by the National Center for Health Statistics. And last but not least, we have the current procedural terminology. Again, this is a coding system, and these are going to be codes for outpatient and office procedures, and this is produced by the American Medical Association. The easiest way to start understanding a little bit more about these terminologies and codes and how they can be used, particularly when you get to some of the computable functions that facilitate reuse, is going to be through the Unified Medical Language System. So we talked about this earlier in the presentation when I talked about how you'd have to set up a UMLS account and log in to get access to the common data elements. The Unified Medical Language System is produced and maintained by the National Library of Medicine, and this is a place that you can see all of the different terminologies. If you wanted to get more information about a LOINC term, if you wanted to get more information about something from RxNorm, you're going to be directed through the UMLS to look at it. What the UMLS actually does is it links together lots of different related terminologies within what they call the meta thesaurus, and they link it to a UMLS concept. So you can't, for example, go into the UMLS and just download the entire SNOMED CT code set and use that in a data plan or in whatever you would be doing to set up a research project, but you can go into the UMLS and learn a little bit more about that code. The last thing I wanted to talk about really quickly was common data models, because this is something that could also go into a data sharing plan. Common data models are likely going to be implemented at an institution level or at a large scale project level. So an individual researcher is probably not going to say, on my own, I'm just going to decide that I'm going to use this specific common data model and that all of my data will map or align to this common data model. But the point of a common data model is to be able to take data from disparate sources, so from different institutions, from different types of studies, and through this common data model layer, be able to transform data so that it can be used very broadly for data collection and data analysis at a lot of different sites. And this slide is the OMOP common data model, which is part of a research network called Odyssey, Observational Health Data something. And the reason that common data models are important is because you're oftentimes with these models dealing with data that are coming out of different health systems, which have their data structured in different ways. So a health system might use a data model that is not necessarily going to match how another health system describes the same condition, the same type of care a patient's getting. There's also issues just in general with lack of standardization. So the two images on this slide show how different sites can represent platelet count units. So for each of these systems, that might be the correct way to do it. But if you try to put all of those together for a common analysis, you're going to have some difficulties with that. The slide on the right shows that you can misspell negative in a lot of different ways, and that can obviously impact your data quality and your data analysis as well. These images both come from a great Coursera course that was put together out of the University of Colorado. So this particular module is called Why Common Data Models. And if you're interested in common data models and how they can help to standardize research, I would definitely recommend checking this course out. The common data models that you're likely to come across within the US are on this slide. So here's Odyssey, which I mangled the name a couple minutes ago, Observational Health Data Sciences and Informatics. You've got the I2B2 Transmart Foundation. PCORNet is a patient-centered clinical research network. If you think back to one of the very first slides with that Advantage trial, that was a PCOR study. And then Sentinel is used by the FDA. And because there's more than one common data model, there's problems now trying to get the data from different data models to work together. So there was a federal project called the Common Data Model Harmonization Project, which worked to build a system where data from all of these different common data models could be integrated for studies. So it had 5 federal partners. They used a bridge standard from the National Cancer Institute, with the outcome being that a researcher can actually put a query through, be able to pull data from a bunch of different common data models, and then have a translated result sent back to them. So the last thing I wanted to do was just leave you with a couple of resources. And again, I hope these slides will be available, and all of the resources are going to be open access. So this is an open access paper out of the Journal of Journal of Biomedical Informatics that talks about some of the problems with fair data sharing specific to common data elements. And one of the big problems, as we saw, is there really isn't one common place to go to find common data elements. So you still run a risk of having a common data element that's been adopted by one study, a common data element that's been adopted by another study, and there may be some middle work that's needed to be able to harmonize that data so that it can be actually used together in aggregate. So this is a good paper. I've got some additional resources here on common data models. So the Coursera course that I mentioned from the University of Colorado is at the top of the page, and this is part of a larger specialization in clinical research and clinical research data, which again is really a fantastic course. And then I just linked to 3 of the common data models that you would probably be most likely to encounter, particularly if you're at a larger institution. Your data have probably been mapped to one of these for participation in the network. And again, in I2B2, the Observational Medical Outcomes Partnership, or OMOP, is linked to the Odyssey Collaborative. And then the Patient-Centered Outcomes Research Network is PCORI or PCORNet. I've got some links here to the UMLS. So they have some great tutorials. This is a really complex resource. There's a lot there, a lot of data and a lot of details, and some limitations on how that data can be used. So if you're interested in more about the specific terminologies, I would refer you both to the UMLS homepage. They've got some great documentation about the source vocabularies that are included in the UMLS, and then they've got a series of tutorials that you can go through to get more acquainted with the resource. And last but not least, I'm linking out to some resources on policy and legislation. So particularly as secondary use of clinical data in research becomes more of a thing, we're intersecting more and more with some of the interoperability requirements that have been coming out from the Office of the National Coordinator, ONC. So what I have here are just some links out to some ONC information on health information exchange and then interoperability requirements that might also be helpful. And with that, I have come to the end of my talk. I've left barely 10 minutes for questions, but I am happy to answer what I can. I will go ahead and stop sharing. And thank you very much for your attention.
Cool. I see a number of people clapping. Do we have any questions at all?
I see Brendan.
I have a question. I have a question. Is there in general a mapping of these are elements that may be personally identifiable information or are always personally identifiable information for the purposes of sharing? Because that seems like something that would be useful to include in these.
Yeah, that's a great question. I think that I've never seen that specifically called out as a value or as something that you can filter on. I would, and again, this is not a clinical researcher, not a clinical research coordinator. I would assume that most of these would be treated within the study as potentially identifiable information because they're always going to be paired with some kind of a case report form that's linking back to an individual. I'm not sure if I'm understanding the question correctly, but I think within the context of clinical research, since these are all going to be referring to an individual, that it would probably be really difficult to tease out just one element and say, oh, you can go ahead and share this, you can go ahead and share the pain scale. Because what does that mean on its own? It's probably going to be more meaningful if these numbers these are the summary statistics for reports from a pain scale from people in this demographic region. And as soon as you start connecting all of those dots back, you're having to think about the fact that some of this stuff could be identifiable.
Because the ultimate goal of this then is to be able to say, okay, well, when we share the data, this is what we're going to share at the beginning. So,
yeah, I think as far as the data sharing plan goes,
I could see that there would be some elements, like I was doing a quick click through, and like some elements would be de facto identifiable.
And I think another thing to think about, that's a really good question. I think another thing to think about is the fact that these aren't necessarily This isn't necessarily data sharing that's going to happen without a data use agreement, that's going to happen without mediation through an honest broker. You know, this isn't the sort of data sharing that lands the content directly into an institutional repository necessarily. You know, what the common data elements really are set up to do is facilitate sharing between research groups or with other researchers that can then use those in further analyses is my assumption. So that may also factor into a data sharing plan in terms of, you know, with appropriate data use agreements in place, we can share XYZ.
Cool. And we have a few questions in the chat. We have one from Brian. Are there preferred structures or formats for the data in these CDMs?
Yes. So Brian, you would probably be one that would be very interested in looking at the actual data models. So each of the common data models is structured somewhat differently, but they all do have their own structures, and some of those will also have values. I actually took out some elements. I had some OMOP, which again is from the Odyssey network. I had some examples of how an OMOP case report might look in terms of, you know, the values and what's being reported and what the notes are, and things like that. So that's where a CDM really, again, is something that probably happens more at an institutional level. So I remember being kind of tangentially part of a group that was working to map a lot of our data for participation in the PCOR network back about 5 years ago. And it was definitely a team effort, and there was a lot of work involved. So that's something probably institutionally or study-wide, as opposed to something that an individual investigator would be thinking about. I hope that answered your question. I saw one from Anna just above it. Are there metadata schemas that are recommended or considered standards for CDEs? My quick answer to that is going to be no, but there's a research group out of Stanford. Let me go ahead and find their website. It's called CEDAR. This is another thing that I actually took out of the presentation because I was afraid it was running a little bit long, but there was actually a paper that this group, the CEDAR group, presented at the AMIA conference, American Medical Informatics Association conference, that talked about kind of a workbench that they had to be able to work with common data elements. So let me pop the CEDAR into the chat, and then if I can find the citation for that paper really quickly, I will plug that in as well. And do I have, so Chris asks, do I have suggestions for how to evaluate different CDEs that one might use? How do you know if a CDE is stable or mature? That is a great question. If you look at the NIH-CDE site, they actually do have a series of annotations. Let me see if I can pull it up, and I don't think I took a screenshot of any of those, but like one of them would be qualified, and that at some level, I think, talks a little bit about some of the maturity that you're referring to. The larger question about how to evaluate is I think kind of tricky because, and that's that paper that I mentioned that talks about how common data elements aren't necessarily used by all of the people that maybe could be using them. That's what makes it a little tricky. Like, do you go for a form that is on the repository that doesn't show any downloads or uses, or do you look at the literature to see if there are, you know, if you can get an example from a paper in the literature that shows a report form where you can maybe extrapolate from that what common data elements were used. So I think that's part of the problem, is that everyone agrees in theory that common data elements are a good idea, but in practice, being able to figure out what the best thing to do for a particular study is, I don't know that there's a ton of guidance on that yet. And I think that that's actually one of the problems with larger adoption. Let me first find that paper that I was looking for, and then I will look really quickly back at the CDE site and see if I can pull quickly that categorization that they use for qualified and other levels that show a little bit about maturity. Okay, so there's the PubMed paper about the CEDAR workbench relevant to the metadata question. And then let's go. There'll be a little bit of a delay here while I sign in. Does anybody have any other questions while I'm waiting for the site to pull up? Okay. I'm going to try and share my screen here. So I was thinking, Chris, of the standard and qualified that you see at the top, and I'm seeing that I was actually wrong. So this actually just talks about how things are managed and isn't anything to do with anything that would show maturity or use or anything like that. What I've done now is just clicked into one of the CDEs just to see if we can see anything in here that might give us any indication. You know, there's some date stuff at the top, but this probably is more administrative data just in terms of when it was added to a repository. And I don't know that you can extrapolate any kind of use from that. Yeah, so at least in this record, I'm not seeing anything that would give me that information. I'm going to look really quickly at that HEAL initiative. Sorry, I know it's super annoying to watch somebody click through pages, but I feel like they addressed this. If I weren't scrolling so fast, you'd be able to see some of my screenshots. They actually did have something on the website about the different levels, which I'm also not finding. Chris, if you want to pop your email address into the chat or just send it to me as a private message, I will look for the thing that I am currently not able to find. I think it was on the HEAL site that they actually had a list of 5 different types or categorizations, and that did refer to a little bit more about where the CDE was in terms of maturity, if I'm remembering correctly. Great, thank you. Copy that. And I see a comment from Brian. I worked with a group that found the NIH recommended CDEs was insufficient to their needs. There's using it, but also some other options. And I think this is a tension that's being felt in a lot of different places. So the work that I'm doing with phenotyping, we're certainly seeing this where, yes, it's great if there can be a common phenotype that's being used in a similar way to be able to identify similar cohorts of patients and set this thing up for comparison. But what's the trade-off between something that really has fitness of purpose for your particular study and then the benefits of the generalizability? And maybe the CDEs are falling a little bit prey to that as well. I see that we are a little bit past time, so thank you to those of you who have stuck it out. Willing to hang out and answer other questions if folks have them, but otherwise, thank you very much for the audience today. It was nice to come and talk about some of this stuff, and good luck preparing for January.
Yeah, and for folks who'd like to see the slides, we can add a link to the slides in the call doc. So I'll email that out again in a week-ish once we have the recording all ready as well.
Thank you so much, Marissa.
Yeah, thanks for having me. It was fun. Deb, for the so when I did the slides, I actually didn't do anything with alt tags for images. Do you want me to add those before I give you the link for the document? Okay. If you can give me day and a half or so to turn that around. I can probably get it back to you by Thursday.
That works.
Okay.